Thanks, Carl. Hello, everyone. I am Abdul Muntakim Rafi. I'm a second year PhD student at DevWorld Lab. I'm someone who believes in dream challenges. I'm someone who believes in dream challenges and their ability to accelerate the field. That's why I feel extremely fortunate that I got to be a part of this. So I think I'm going to talk about the data first. So the participants were given a set of around 6.7 million promoters sequences and corresponding expression values to train their models. And so these sequences were, the expression for these sequences were measured in a high throughput experiment. So they were a bit noisy. So obviously we could not use these sequences to evaluate the models. So that's why we did a second experiment to create the test data set where the test sequences were designed and each of the sequence were measured multiple times. And so that's how we were sure that, okay, this is a better quality data. And these sequences were designed in a way that they have biological relevance. For example, a lot of them had SNPs. I'll get to that later. So what is the goal of this challenge? That you train your networks with millions of random promoters, which are pretty noisy, but you would evaluate your models on sequences which are biologically relevant. Now, in this competition, we had a set of important rules. The first one is that, okay, we did not allow the use of external data because we wanted to make sure that all the participants were training their models with the same set of sequences. And the second rule was that, okay, ensembles were not allowed. So I think because of the spirit of this competition, that we wanted to see, like, what are the best network architectures or training strategies for genomic sequence data. So if we allowed ensembles, maybe we'd have gotten better results, but this question would still remain unanswered. So that's why we had this rule. And the third rule is that, okay, pseudo leveling of the test set was not allowed because we did not want the challengers to use the test set in any way. And pseudo leveling is the trick that calculators use to make predictions on the test sequences and then feed it back to the network. And it's a weird thing, but works. But so these things we did not allow in our competition. And let's look at the overview of the test data set. So the test set had around 71,000 promoter sequences, and they belong to 8 different promoter classes. And we had promoters with high expression, promoters with low expression. We had promoters taken from the yeast genome, and there were a couple of sequences that are totally random. We chose some sequences where the previous version of the models that I think Carl developed, those models struggled on those sequences. So we included those. And we had also a lot of sequences where the sequences only differed by a single base, or also sequences where the transcription factor binding sites were perturbated, and also a set of sequences where the transcription factor binding sites were tiled across the sequence. And we checked two things: that how good the participants are at predicting the exact expression, and how good they are at predicting the change in expression. Now to the competition metrics, we used 2 metrics to evaluate the models, score Pearson R-square and score Spearman. These are just the weighted average of the Pearson R-square and Spearman values across all test subsets. And we used different weights for different test subsets according to their relevance to the field, and also how difficult it is. We thought that, okay, how difficult it would be to predict those particular test subsets. And you can look at the weeks here. Now, onto the evaluation. So we had two stages of evaluation. We had the public leaderboard stage and the private evaluation phase. In the public leaderboard phase, 110 teams participated, consisting of like 300 participants from all around the globe. We are actually people from all the continents except Antarctica. And so 13% of the test set was used in this stage for evaluating the models. And so what would happen is that people would submit the predictions in a website, and we would just tell them, okay, what is their score Pearson R-square and score Spearman score. But we did not tell that, okay, what was the performance for different test subsets. So because this was just a way to actually give an idea of the model's performance without telling too much information, so that there is no way that, okay, you can start overfitting on the test sequences through this stage. And in the private evaluation phase, that was the next round where the top performing teams actually submitted their code base, a report detailing their approach. And at this stage, we used the rest of the test data. Now, I think this is an interesting slide. I think this shows the progression of the performances of the different teams. So I included only the top 10 teams here. So you can see that how, I think, Unlock DNA, who is here today, high schoolers are all you need. And I think there's another thing that, okay, their performance actually significantly improved throughout the course of the competition. So I think this also shows, okay, why challenges like this actually, I think, help us grow and build just better things. And this is also, I think, a similar trend. Like this is just a progression in score Spearman across different teams. So at the final evaluation stage, so we used bootstrapping. We chose 10% of the remaining test data, like n number of times, and we ranked all the teams in each of the pools, and the teams with least sum of ranks in this, like, n pools were considered to be the better performing, and we ranked them according to scores Pearson R-square and score Spearman separately. Okay. I think, yeah, from this illustration, actually, you can see that, okay, how the teams fared in this all these n number of pools of the test data. You can see that, okay, AutoZone were actually in the first position in all of the pools in the bootstrapping, and DHA also was visibly in the second place, and Unlock DNA was in third place. And also for some models, you can see, okay, they're pretty like much head-to-head each other. So after we did this, then we wanted to verify that, okay, the models that were submitted to us, like, okay, they are reproducible. So from the previous graph, where actually it's kind of visible that, okay, the top 3 teams were significantly ahead of others. So we went on and just implemented their submitted models. So in this part, we ran their whole code from scratch, and we were able to reproduce their claimed performance levels. So I must thank the challengers because they submitted codes which are pretty much well documented, and it was really easy to just run it. So I would like to present to you the top performers of this competition. The top 3 teams will be presenting their solutions today, and the other teams will be presenting their solutions in the poster session. And I would like to a huge shout out to high schoolers are all you need team. It's the team literally made of high schoolers, and I would like to send my appreciation to their supervisor Peter Ko for investing his time in developing the next generation of scientists. I'm excited to see like what they develop in the future. And I'd also like to thank Google TRC team because they worked really hard to provide computational resource for all the teams. And I think that made our competition equitable for every team. Anyone could actually build models like deep models and train them. Just to give a perspective, if we were to pay for the computational resource that TRC gave us, we'd have to spend millions of dollars.